particular webinar has been broadcast a little more broadly to members of the Pistoia Alliance, to ChemAxon user group, and also to the SLA chemistry division. So we're pleased to bring you this series and pleased that you've joined us today. One of the motivations was to extend the work that the programming committee of CIMF does in programming at the national meetings. And there actually was a presentation in Indianapolis earlier this month about HELM from Barry Bunin. But we're pleased to have with us today Roland Knispel from ChemAxon, who's been involved in the technical group within Pistoia dealing with HELM. We appreciate your feedback. Chair at acscimf.org if you have comments or suggestions of additional webinars, different topics that you'd like to hear. And so just a word about Adobe Connect. If you're not in full screen mode, you should see a question box or a comment box, and if you type something there, we'll use it excuse me, to ask questions and hopefully have Roland answer those later on. If you're having technical difficulties, also feel free to enter something in that box, and I'll try to address that. I'm getting some queries about sound. Sorry, I don't have a telephone dial-up line. Let me see if I can enable one. Whoops. Sorry, I may have disconnected a couple people there. Sound streaming okay, but if I speak a bit quieter, we'll try that. Okay, so again, keep typing those technical problems in, and we'll see if we can address them as time goes on. So this was a note to me. I've already started the recording, and we'll post that later. And as I said, we're pleased to have Roland Knispel with us today. HELM for the masses, a notation standard for representing biologics. A little bit of information about Roland. He has a doctoral degree from the Technical University of Munich. He's had educational background spanning theory and wet chemistry, and so really in an ideal position to be involved with the Pistoia Alliance as well as with the HELM Project. So with that, I will turn it over to Roland. And Roland, you should be live now.
Hello.
Share your screen.
Yes. Can you hear me well?
I can hear you fine.
Okay, so I'm just going to share my presentation with you. And also from my side, a really warm welcome to today's webinar. I'm going to talk about the HELM, a rather young notation standard for the representation of biological macromolecules. HELM has been originally developed by Pfizer Research Informatics Group as a means of handling the data about biological macromolecules within their company registration systems. And the transformation of HELM as a proprietary technology at Pfizer towards an open standard in the public domain has been the main aim of one of the recent projects of the Pistoia Alliance, in whose name or on whose behalf I'm talking to you today. So I would like to start off with why such things happen, give you a brief introduction about what is the Pistoia Alliance. The Pistoia Alliance is a formally established cross-company organization with the general mission to lower innovation barriers of R&D in their member organizations by improving the business processes of their research and development, and also facilitating the interoperability of R&D operations across the different partner organizations. By fostering this collaboration in the pre-competitive space, all member organizations equally benefit from the efforts taken within this Pistoia Alliance. On this slide, you can see, as of the second quarter of this year, I think it's not even an exhaustive list of companies who are members of the Pistoia Alliance. There are the major pharmaceutical companies represented in the board, which determines strategy and directives of the Pistoia Alliance, selecting projects and defining areas where to get involved. However, all of the additional members, which are listed below that, do have a say and regularly meet in kind of strategic meetings in order to identify areas of interest common to the member consortium. The HELM project, of which I'm going to talk to you about today, was initiated based on a commonly identified need across the consortium members. And this project actually was started pretty recently, just about 10 months ago. And it states in the project charter that at present most pharma companies have no way of storing complete structural, machine-readable information about the macromolecules they prepare or use in their research projects. Sorry, I was just listening to a question from Sergio. The business case the Pistoia Alliance took was essentially driven by savings in productivity by adoption of this HELM notation. So example given that fewer notation queries or resolutions from non-standard language annotations of the macromolecules arise, that it may be serving as a kind of standard reference notation for all macromolecules in pharma compound registration systems. And it was thought that with its large span over vendors, pharmaceutical companies, and other information management related companies, the Pistoia Alliance would be the best positioned organization in order to achieve this game. The value proposition of HELM is that, well, HELM has been initially developed, has been tested, and is used at Pfizer for about 4 years now. HELM is something which can benefit from not only being proprietary to the limited audience of the consortium members, but we actually see it as beneficial if this is openly published and supporting software tools are available to the larger chemoinformatics or bioinformatics community. Hence, there were efforts in order to publish this in the open source domain. And the Pistoia Alliance, with its kind of larger reach, is just perfect to signpost this standard and foster its adoption not only throughout the Pistoia Alliance community, but also as a speaker towards other institutions such as legal authorities, such as public domain databases, which inherently have trouble in kind of managing their informations about large molecules, where you see more and more convergence between small molecule world and the world of biological macromolecules. So what is HELM? HELM is the abbreviation for Hierarchical Editing Language of Macromolecules. And as such, it is a mean to represent non-small molecules, or often termed biologics, in a language which formally contains a notation system, which means it defines how it is actually transcribed into a string. And it comes with a grammar and a vocabulary, how these sorts of strings are written out. It is hierarchical because the individual elements of a HELM representation start from the very bottom using regular atoms and bonds, which are constituting monomers. Monomers have additionally defined data, for example, attachment points of how these monomers can interconnect with each other. And some interconnected monomers, in turn, can build simple polymers. which are linear chains. And a number of such simple polymers then finally constitute a large macromolecule, what we call a complex polymer. Let's go into detail. So I said that in the beginning, atoms and bonds are the basic structure of it. The monomer itself, as you can see, has a chemical structure with some attachment points defined, and it comes with some additional data which defines its role within the representation of HELM. So for example, if I'm looking here at the structure of alanine, then I identify the typical attachment points for building the peptide backbone on the amine function and the carboxy function with R1 and R2. I define a so-called natural analog. This is for compatibility reasons, with generating faster compatible sequences of monomers which carry modifications, for example. I define the type of the monomer I'm looking at, so I'm making sure that a simple polymer contains only amino acid residues, for example. And finally, I can add a name identifier to it. On the simple polymer level now, I'm constructing out of monomers of a given polymer type a linear chain of all monomer units, which is the backbone. And sometimes in case of nucleic acids, I also attach a branch monomer. So for example, if I'm looking at RNA or DNA molecules, then I do have the ribose and phosphate individual units as monomers. However, only those build the backbone of the entire nucleic acid sequence. The branches are then the corresponding nucleobases. That looks like this as an example. So if we are going to convert a regular sequence AUG of RNA type into a HELM notation, then this would be the corresponding simple polymer notation for this sequence. You can see that the R and P correspond to the phosphates and the ribose. And in brackets, because it is an attached branch monomer, we use the uracil. Now I can, of course, introduce non-natural monomers into such a sequence, and this is the real strength of the HELM notation after all. So if you had, for example, a methylated ribose, and you would use a thiophosphate in order to stabilize your molecule against hydrolysis, then you can have additional monomers identified in your dictionary which describe exactly this, and the notation would use the abbreviations defined for the individual monomers. So this is here, in this case, the version what you see over here on the right-hand side. For complex polymers are essentially the entire macromolecule and contain as such the entire structural information about it. It doesn't only carry atom-to-atom bonding information, but HELM annotation also allows you to kind of add additional data in a separate section of the HELM notation itself. I'm going to explain this now on a little example. So the HELM notation and schematically looks like what you see here up on the top left side. You have 4 sections within that notation. The first section identifies all simple polymers which the macromolecule you are describing is comprised of. Within simple polymers, you know that the connections between the monomers are predefined by certain rules. However, if you want to describe connections between individual simple polymers, they would be listed in the second section, the list of connections. The third section contains a similar concept for the base pairing or hydrogen pairing of, for example, double-stranded DNA molecules, where each hydrogen pair essentially relates to a hydrogen pairing between the interacting nucleobases of the 2 strands. And the last section is a list of annotations, where the annotations refer to individual simple polymers of the macromolecule. If we formalize this, then it looks a little more complicated. So we are looking for certain polymer types and assign running numbers in order to identify the list of simple polymers. And within these curly brackets, you then put the simple polymer notations, which essentially is the list of monomers separated by dots. And with pipe symbol, you separate one simple polymer from the other, and you can add as many as you want. For the list of connections, we have to identify from where originates a connection and where does it bind to. So we look at the source polymer ID and target polymer ID, and then identify at the corresponding position of that simple polymer which attachment points to include for this bond. The third one is largely similar to the list of connections, except for that we do not specify a specific attachment points, but we use the keyword pair just in order to somehow describe that this is a base pairing. And with the same polymer IDs which you have given to simple polymers in the first step, you refine the description about your molecule by giving additional annotations to individual simple polymers. With a real example that would look like this. So you have here an RNA moiety. The individual functional units are separated by dots. You have an additional peptide, of which the sequence is clearly identifiable here. And there's a PEG3 linker, which is a single monomer leveraged to the level of a simple polymer. And then you have the second part, which identifies the connections. So the Chem1 is the linker molecule, and it appears in both of the identified connections. And the RNA is bound to the second attachment point, or first attachment point, and the peptide is bound to the second attachment point of the chemical linker in between them. There are no hydrogen pairings in this molecule, since it's only a single strand of RNA. And in the fourth section, I could, for example, identify that the RNA strand type is that it's single-stranded. Now, there is a specification of this, and there is a publication on this. And both of them you can find online on the openhelm.org webpage. I will mention this later. What I want to focus on right now is what kind of tools are actually available for you if HELM is interesting to you, or you would like to assess how HELM could be useful for you, and what sorts of things can I build with that. So the HELM is implemented in two separate projects currently. One of them is the HELM Notation Toolkit, which handles all of the grammar and vocabulary and monomer management related to the implementation of HELM. And simultaneously, there exists a graphical user interface, the HELM editor, which in the background uses the API of the HELM Notation Toolkit in order to facilitate graphical editing of macromolecules conforming with the notation itself. Then also the editor, though, has certain functionalities which would allow you to call this as an API. So we use it, for example, internally by injecting HELM notations into the canvas of the editor. So you actually have quite large control about how to control the editor itself. And what you see here on the right-hand side is actually a screenshot of the editor. It has some tool buttons, a panel on the left-hand side, where the individual icons you see are graphical representations of the monomer stored in the current monomer dictionary, which the editor reads. And you have here on the right-hand side a drawing canvas on which you can drag individual elements from these building blocks and then start dragging in order to build the connections between them. But you see also here a little text box up there where you could directly import a final HELM notation string or a peptide sequence or nucleotide sequence. I do not want to go into too much detail here because we have some really nice tutorial videos on the openhelm.org webpage. What I want to pinpoint here is that both of these individual components, the editor and the notation toolkit, are freely available as open source projects, and they are hosted at github.com. You see the link here. If you visit there, you will find the HELM editor and HELM notation toolkit projects. Now, what can we do with these tools at the moment? The original research article has a number of examples. Some of them are very simple, some of them are a little more advanced. I decided to skip over the very simple ones. Obviously, you can just display a peptide sequence. This is here a tetraloop where you have a single RNA strand which kind of folds onto itself. And if you identify the base pairings between the corresponding and matching nucleobases and have the other ones kind of unbound to anything, the resulting HELM notation would be this. And I'm just going to show you a few more examples here. If we are looking at a conjugate where you are binding a single RNA sequence to a peptide sequence, for example, you have your messenger RNA tag of your protein covered by a specific chemical linker, then you would be able to express this, and this is the corresponding HELM notation for that. We at Chemaxon, we also made some investigations about how could we possibly treat glycoproteins, because conceptually the only defined polymer types the notation currently handles are RNAs and peptides, where RNAs obviously cover DNA and RNA at the same time. If you take the work and kind of identify all of the appearing sugars as chemical modifiers, then you would well be able to kind of represent this molecule within the HELM notation. However, it has the shortcoming that you couldn't directly access the kind of sugar sequence out of this notation, since each individual sugar is a separate component, and you would have to look up in the connection table in order to identify where is my glycane chain, which I'm interested in. Something I was just seeing while I was preparing these slides is look at the first 4 amino acids, and if you rearrange the letters slightly, it actually shows HELM. I found it really funny when I saw that today. It's a sign that it's actually designed for exactly this case, right? Now, the aim of the Pistoia Alliance HELM project was to provide a standard reference notation system for macromolecules in order to use them for compound registration systems, especially in the pharmaceutical industry, but also in other industry areas. Now, there are a set of requirements which need to be matched in order to do so. And first of all is like, okay, does it cover the use cases which are of current interest? The second, is it specified well enough and is it documented well enough that people can readily and easily integrate this into their system architectures? The third point is the question of interoperability, which means now at one organization I may happily implement it and everything runs smoothly. However, as soon as I need to transfer information about the macromolecules I'm working with to an external partner organization, for example, am I able to easily transmit my information to that partner company and they have no trouble in kind of picking up the work from that? The fourth point is registrability. Obviously, if it's about to be used in registration systems, what are the efforts in order to be able to register molecules via their HELM notations? The fifth point is about the stability. Obviously, if there is a major architectural change required in my organization, then do I have the confidence that this standard will be maintained for more than just 2 years? And finally, adoption. So adoption in the end drives everything a little bit. If the previous 5 requirements are not met, then it's not very likely that the HELM standard itself will be adopted. Let's look at all 6 points in the order of their appearance here. Regarding use case coverage, so we've seen that HELM notations can rather easily represent anything which has a simple nucleic acid or protein sequence. So it's covering things like regular DNA, small inhibitory RNA, proteins, antibodies. But it's also able to represent where sequences are simply not sufficient, modified biological sequences. So when you introduce locking, for example, in your DNA or RNA molecules, when you have proteins which are carrying post-translational modifications or other chemical modifiers, then this is a situation when HELM can be applied and all of your chemical structure information is maintained. We've seen that in principle it also applies for glycoproteins. However, it would be advisable to add better support for the polysugar component as a polymer type, which is currently not implemented. With respect to conjugated molecules, which are combinations or hybrids of peptides and RNAs, we have seen one example which can be expressed. There are quite fashionable molecules, antibody-drug conjugates. Now, what's the situation with those? The problem with antibody-drug conjugates is that they are created usually by applying a conjugation reaction, which leads to a set of molecules. Like you have the core of the antibody, heavy chains and light chains, and you create a quite large variety of individual molecular structures with different patterns of the conjugation. This is something which currently is not well covered within the HELM notation, but it is something which we are obviously looking within the Pistoia Alliance as one of the closer in the future enhancements in order to make that suitable for these sorts of cases as well. Specification and documentation. The HELM project has its own webpage. It's called openhelm.org. See the link down here. On this we are collecting all sorts of information which may be relevant for the public domain. For example, you have here on the right-hand side a tryout button, which leads you to a page where the HELM editor, of which you've seen a screenshot, opens up in a browser window as a Java applet, and you can start playing around with the editor and explore a little bit what it does for you. And if you click on the HELM notation tab, it will lead you to a kind of collection of resources where we provide tutorial videos in order to get familiar with the editor. It contains a recording of a webinar which Tianhong Zhang, one of the kind of developers of the HELM notation toolkit, has been recorded, giving a little guidance of the open source repositories on GitHub, how to use the code, where to find certain things. There was the question on the Chemint mailing list today about why isn't the specification online. I've responded to that list, and I unfortunately have to tell you that technically the specification in version 1.0 is finalized. However, it still needs to go through approval processes, but I expected that by the beginning of October it will also be linked here on that webpage. Interoperability is the thing. So there are two situations I would like to draw your attention to. The first one is the situation as it happened at Pfizer, where a single organization uses HELM and they have a monomer administration which essentially is centralized. So you can input anything, be it a HELM string, be it a protein or RNA sequence. It goes into the notation toolkit, and then the toolkit itself contains function which readily convert this into SMILES representations or multiplies, which allow you to, if you really do not want to build a HELM-specific registration system, to easily link this to your compound registration systems you have in-house. On the other hand, with the additional parameters of the so-called natural analogues I have mentioned for the monomers, you also have kind of a bridging access to any sort of biological sequence management tools you may apply. And this is a back and forward communication, which in the end, via the HELM notation toolkit, leads to an indirect communication between your compound registration and sequence management, which is valuable in the case you're looking at these sorts of chemically modified conjugate molecules. The reverse direction, having structures registered in your compound registration to the HELM notation toolkit, is something which is not entirely solved, but it's not impossible at all. The easiest kind of application would be if your compound registration uses a representation which, for example, allows superatom group definitions. Then the HELM notation toolkit could parse out these superatom groups, interpret them as the corresponding HELM monomers, and then this sort of thing is facilitated. The second thing, and I think a few vendors are actively working on this, is to actually try to perceive the biological information contained in a plain chemical structure. That you have a small peptide as a SMILE string and try to identify which amino acids are actually building up that SMILE string. And then you have, again, the opportunity to build a HELM description from this molecule. The kind of second point is you would like to exchange this sort of information with somebody else, then you have to be aware of that currently the editor is only capable of exporting the HELM notation string itself, but it is not able to currently encode the monomer it is using in order to include this in this exchanged file. So in order to regenerate the entire atomic structure, obviously the monomer information has to be passed on somehow additionally. This is, I think, the highest priority area the Pistoia Alliance will take as a next step in order to provide this sort of interoperability. When you're looking at kind of building a system which registers your macromolecules based on the HELM notation, then there's one additional bit which also currently has serious shortcomings, let's say, which is the canonicalization of a HELM notation. If such a canonicalization would be robust enough, you can imagine that you can directly register all of your molecules in the HELM annotation. And essentially, you could think about eliminating individual compound registration and sequence registration tools and have a set of services which kind of facing towards the rest of the infrastructure just generates on the fly or upon request corresponding sequences or biological structures. The registration of HELM monomers, however, since you can imagine that different organizations applying HELM and there is nothing centralized, that the Pistoia Alliance goes ahead and says, look, we are the single authority which defines which monomers are available to you. It obviously doesn't make sense, is to consistently register these sorts of monomers. Imagine the case that you have a phosphorylated histidine here, then you could think about that for some applications, it's enough just to register that as a single monomer. However, depending on the context of questions you'd like to ask further downstream, it may be wise to actually state that, look, this is a histidine residue which carries an additional modifier, because that allows you to make much easier queries like which of my residues carry a specific phosphorylation on one of the two possible sites of the histidine. If you're just looking at the residue itself, these sorts of queries are a lot more complex and will take a lot more time to be realized. About long-term stability, I think I've made the point largely in the kind of description of the business case. So we have a strong incentive within the Pistoia Alliance to somehow signpost and speak on behalf of a rather large consortium of industrial and vendor partners. The Pistoia Alliance has agreed that it would provide some sort of an ad interim governance for a controlled evolution of the standard, depending on the relevant use cases of interest to the community. And we have also released the HELM tools to the open source public domain in order to facilitate a public discourse. This now should encourage everyone that you find shortcomings in the notation toolkit, in conceptual flaws of the notation itself, or in the editor, to come back and either improve it on your own or write requests to the Pistoia Alliance or the project itself in order to see whether this can be meaningfully and technically included in the next version of the HELM notation standard. Obviously, because it's a rather young standard, we do not see widespread adoption yet. But on the other hand, we obviously have one large pharma company who is applying this productively. We have a few vendors which are currently implementing tools handling export and import of HELM notation, and I'm sure there will be a number of applications to follow on this line. And we are also seeing a number of pharmaceutical companies and even public institutes such as the EBI showing significant interest because they would like to evaluate how HELM notation could potentially help administrating their information management on macromolecules. That brings me to the adoption slide. I've shown that to you. We've had communications with a few other kind of organization groups such as content providers. We could imagine that scientific publishers, for example, adopt HELM in order to facilitate providing a consistent notation for the description of larger molecules in scientific publications. Immediately, academia would profit from this. State agencies such as the FDA, they have a large set of regulations for approval of biological biotherapeutics, if you want, and they are seeking to formalize the way how you submit the molecular information or the structural information about these molecules. That said, the standard is young and requires probably a thorough assessment whether it is really a robust tool, but we strongly believe in that it is robust enough because it covers a lot of the use cases relevant right now, and it's considerably easy to extend it because of its modular nature. So in summary, I would say that the HELM notation is probably already meeting most of the requirements for broader adoption. And what I would like to state here is that regarding specification and documentation, interoperability, registrability, I think we have a very, very broad coverage of things. Regarding use cases, there may be specific organizations who say that for us this is not very interesting unless specific additional features are included. And regarding long-term stability, I would say this is largely a fact which is strongly connected to the level of adoption of this HELM notation standard. And this is something which obviously you as the audience here now have the kind of playground in order to see whether we can leverage this in order to increase and optimize information management of larger biological macromolecules. So if you're looking for more information, then please visit this webpage, openhelm.org. If you have any suggestions or you want to get involved with the project or the Pistoia Alliance, feel free to contact us via email at info@openhelm.org. With this, I would like to conclude my webinar for today. And I think, as far as I know, Sergio Rothstein, who is the domain lead, would also be available for audio, and we can have our Q&A session now. I just need to switch back to the management screen.
Yeah, thanks, Roland. We do have